Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The dawning of recombinant deoxyribonucleic acid technology in 1973, principally achieved through the collaborative efforts of Stanley, Cohen and Herbert, represented a watershed moment in the biological sciences. Their groundbreaking work, detailing a method for the construction and propagation of biologically functional plasmids in Escherichia coli, propelled molecular biology into a new era, offering unprecedented opportunities for gene manipulation and expression. This essay undertakes a comprehensive evaluation of the core claims presented within their seminal publication, examining the methodology, evidence, and implications of this transformative research. We will dissect the arguments advanced by Cohen and Boyer, contextualize them within the prevailing scientific landscape, and assess their enduring impact on the trajectory of biological Section 1 Introduction and Background. Cohen and Boyer's 1973 publication outlines a novel methodology for the creation of recombinant deoxyribonucleic acid molecules capable of replication and expression within a bacterial host. The work hinges on the ability to cleave deoxyribonucleic acid molecules at specific sites using restriction endonucleases, join these fragments with compatible ends, and introduce the resulting recombinant molecules into E. coli cells, where they are subsequently propagated. The authors persuasively argue that this approach allows for the transfer and amplification of genetic information from diverse sources, providing a powerful tool for the study of gene function and the production of valuable biological molecules. The broader context surrounding this publication includes the burgeoning field of molecular biology, driven by advances in deoxyribonucleic acid sequencing, plasmid biology, and bacterial genetics. The discovery of restriction enzymes, in particular, provided a crucial tool for deoxyribonucleic acid manipulation, setting the stage for the development of recombinant deoxyribonucleic acid technology. This initial exploration provides the foundation for a systematic evaluation of the pivotal ideas proposed by Cohen and Boyer. Section 2 Cleavage and joining of deoxyribonucleic acid molecules. At the heart of Cohen and Boyer's methodology lies the capacity to precisely cleave and join deoxyribonucleic acid molecules. They demonstrate the use of the EcoR1 restriction endonuclease derived from Escherichia coli strain RY13 to generate specific deoxyribonucleic acid fragments with cohesive, single-stranded termini. They explicitly state the sequence specificity of EcoR1, highlighting its ability to recognize and cleave deoxyribonucleic acid at the sequence guanine, adenine, thymine, cytosine, producing fragments with complementary adenine, thymine overhangs. The authors then employ deoxyribonucleic acid ligase to covalently join these fragments, exploiting the complementary nature of the cohesive ends to facilitate efficient ligation. This is a fundamental claim, as the precise and predictable cleavage of deoxyribonucleic acid molecules is a prerequisite for the controlled construction of recombinant molecules. The evidence presented in support of this claim includes gel electrophoresis data demonstrating a specific cleavage of plasmid deoxyribonucleic acid by EcoR1 and the subsequent joining of the resulting fragments by deoxyribonucleic acid ligase. The efficiency and specificity of this cleavage and ligation process are critical for the success of the overall recombinant deoxyribonucleic acid methodology. Cohen and Boyer convincingly argue that this precise control over deoxyribonucleic acid manipulation is a key element of their innovation. Section 3 Construction and transformation of recombinant plasmids. The next pivotal step involves the construction of recombinant plasmids and their subsequent introduction into E. coli cells. Cohen and Boyer describe the creation of a recombinant plasmid, PSE101, by combining fragments of the E. coli plasmid coli-1 and the Staphylococcus aureus plasmid PSE101. Plasmid PSE101 confers resistance to the antibiotic tetracycline, providing a selectable marker for transformed cells. The authors then describe a transformation protocol involving the treatment of E. coli cells with calcium chloride to render them competent for deoxyribonucleic acid uptake. The successful transformation of E. coli cells with the recombinant plasmid PSE101 is demonstrated by the acquisition of tetracycline resistance. The claim here is that foreign deoxyribonucleic acid can be stably introduced into a bacterial host via a plasmid vector, conferring new traits to the recipient's cell. This is a crucial aspect of the technology, as it allows for the transfer and expression of genes from diverse sources in a convenient and well-characterized host organism. The evidence for this claim includes the observation that transformed E. coli cells exhibit tetracycline resistance, indicating the successful incorporation and expression of the PSE101 plasmid deoxyribonucleic acid. Controls, such as transformations performed without plasmid deoxyribonucleic acid, confirm that the observed tetracycline resistance is due to the presence of the recombinant plasmid section for replication and expression of recombinant deoxyribonucleic acid in E. coli. Having demonstrated the construction and transformation of recombinant plasmids, Cohen and Boyer proceed to assess their replication and expression within E. coli cells. They show that the recombinant plasmid PSE101 replicates autonomously within the E. coli host, maintaining its integrity and stability over multiple generations. They also demonstrate that the tetracycline resistance gene encoded by PSE101 is actively expressed in the transformed cells, conferring resistance to the antibiotic. This establishes the functional competence of the recombinant plasmid within the bacterial host. The key claim here is that the transferred genetic material is not merely present but is also actively replicated and expressed, ensuring its stable inheritance and functional impact on the host cell. This is a significant advancement, as it demonstrates the potential for using recombinant deoxyribonucleic acid technology to engineer bacteria with desired traits or to produce valuable biological products. The evidence supporting this claim includes measurements of plasmid deoxyribonucleic acid copy number in transformed cells and assays of tetracycline resistance levels. These experiments confirm that the recombinant plasmid is stably maintained and actively expressed in the E. coli. Section 5 Stability and inheritance of recombinant plasmids. A critical aspect of the Cohen and Boyer methodology is the stability and inheritance of recombinant plasmids in bacterial populations. The authors present data demonstrating that the recombinant plasmid PSE101 is stably maintained in E. coli cells over multiple generations in the absence of selective pressure, for example tetracycline. This indicates that the plasmid does not rapidly lose its integrity or get eliminated from the population. They also investigate the inheritance of the recombinant plasmid during cell division, demonstrating that both daughter cells inherit the plasmid, ensuring its continued presence in the bacterial culture. The central assertion here is that the recombinant deoxyribonucleic acid is not merely a transient guest within the host cell, but a stably integrated component of its genetic makeup, capable of being reliably passed on to subsequent generations. This is a crucial factor for long-term applications of recombinant deoxyribonucleic acid technology, such as the production of recombinant proteins or the genetic modification of organisms. The evidence for this claim includes experiments tracking the presence of the PSE101 plasmid in E. coli cells over time under non-selective conditions. The continued presence of the plasmid, as assessed by deoxyribonucleic acid hybridization and tetracycline resistance assays, demonstrates its stability and inheritance. Section 6 Broader applicability and future directions. Cohen and Boyer conclude their publication by discussing the broader applicability of their recombinant deoxyribonucleic acid technology and outlining potential future directions. They suggest that the methodology could be used to transfer genes from diverse organisms into E. coli, allowing for the study of gene function and the production of valuable biological molecules. They also acknowledge the potential risks associated with the technology, such as the accidental release of genetically modified organisms into the environment, and emphasize the need for careful regulation and ethical considerations. The main argument put forth in this section is that this technology opens up a vast landscape of possibilities while simultaneously underscoring the importance of responsible development and application. The authors do not shy away from recognizing the potential downsides, demonstrating intellectual honesty and a commitment to ethical scientific practice. This section serves as a powerful call to action, urging the scientific community to explore the potential benefits of recombinant deoxyribonucleic acid technology while remaining mindful of its potential risks. The future directions they suggest, such as transferring genes from diverse organisms, would come to fruition in the following decades, transforming biotechnology and Section 7 addressing potential concerns and limitations. While the Cohen and Boyer paper elegantly demonstrates the feasibility of recombinant deoxyribonucleic acid technology, it is important to consider potential concerns and limitations inherent in the early stages of this field. One potential concern is the possibility of unintended mutations arising during the construction or propagation of recombinant plasmids. The authors acknowledge this risk and emphasize the importance of using well-characterized strains of E. coli and carefully controlling the experimental conditions to minimize the likelihood of mutations. Another limitation is the relatively low efficiency of transformation in E. coli cells. The authors recognize this limitation and suggest that further improvements in transformation techniques would be necessary to enhance the efficiency of the methodology. A further point to consider is the dependence on EcoR1, the specific restriction enzyme used during the experiment. The authors acknowledge other restriction enzymes exist. The field would later benefit from hundreds of restriction enzymes, which allow scientists to cut deoxyribonucleic acid at difference. The claim that the authors are making in this section is tacitly that while technology is not perfect, the benefits outweigh the costs. The authors attempt to address potential concerns and limitations by addressing them and providing solutions, acknowledging and accounting for any issues. Section 8 contextualizing the work within the scientific community. The 1973 publication of Cohen and Boyer's work on recombinant deoxyribonucleic acid immediately resonated within the scientific community, sparking both excitement and trepidation. The ability to precisely manipulate and transfer genes opened up unprecedented avenues for research in molecular biology, genetics, and biotechnology. However, the potential risks associated with the technology also generated considerable debate and concern. Scientists and policymakers grappled with the ethical and societal implications of recombinant deoxyribonucleic acid technology, leading to the establishment of guidelines and regulations to govern its use. The Lysomer Conference on Recombinant Deoxyribonucleic Acid in 1975, for example, brought together scientists, ethicists, and policymakers to discuss the potential risks and benefits of the technology and to develop guidelines for its safe and responsible use. The claim that the scientists were making here is by releasing the paper, an attempt to generate awareness and discussions on implications of the technology in the scientific community. This ultimately resulted in discussions of ethical implications and potential harm of the technology. Section 9 Subsequent developments and refinements. Following the publication of Cohen and Boyer's seminal work, recombinant deoxyribonucleic acid technology underwent rapid development and refinement. New restriction enzymes were discovered, increasing the versatility of the methodology. Improved transformation techniques were developed, enhancing the efficiency of gene transfer. New plasmid vectors were constructed, providing a wider range of options for cloning and expressing genes. Furthermore, Techniques for sequencing deoxyribonucleic acid were invented. These included Sanger sequencing and the Max-Ham-Gilbert method. These advances collectively transformed recombinant deoxyribonucleic acid technology into a powerful and widely used tool in biological research. A central argument of the section is that the initial publication by Cohen and Boyer served as a catalyst for subsequent innovations and improvements in the field of molecular biology. One can see that this pivotal contribution helped pave the way for a new era of genetic engineering and genomic exploration. Section 10. Applications in biotechnology and medicine. Recombinant deoxyribonucleic acid technology has had a profound impact on biotechnology and medicine, leading to the development of numerous valuable products and therapies. Recombinant deoxyribonucleic acid technology has been used to produce a wide range of therapeutic proteins, including insulin, growth hormone, and erythropoietin. These proteins are produced in large quantities in bacterial or eukaryotic cells and are used to treat a variety of diseases. Recombinant deoxyribonucleic acid technology has also been used to develop new vaccines against infectious diseases. For example, recombinant vaccines against hepatitis B and human papillomavirus have been successfully developed and are widely used to prevent these infections. In addition to these applications, recombinant deoxyribonucleic acid technology is also being used to develop gene therapies for genetic disorders. Gene therapy involves the introduction of a functional gene into a patient's cells to correct a genetic defect. The main claim is that the basic principles outlined by Cohen and Boyer have been translated into tangible benefits for human health and well-being. Recombinant deoxyribonucleic acid technology has revolutionized the pharmaceutical industry and opened up new avenues for treating previously incurable diseases. Section 11. Ethical considerations and societal impact. The development and application of recombinant deoxyribonucleic acid technology have raised a number of ethical considerations and societal concerns. One concern is the potential for the technology to be used to create genetically modified organisms that could harm the environment or human health. Another concern is the potential for the technology to be used to discriminate against individuals based on their genetic makeup. These concerns highlight the importance of careful regulation and ethical oversight of recombinant deoxyribonucleic acid technology to ensure that it is used responsibly and for the benefit of society. The ethical concerns continue to evolve as technology rapidly moves forward. Similar to Section 6, the Cohen and Boyer papers serve as a catalyst toward generating many discussions on the benefits and harm of the technology. The claim scientists are making here is by having the discussion in the first place, they are mitigating any risks of unforeseen danger. Section 12. Conclusion. Enduring significance and future prospects. The 1973 publication by Stanley Cohen and Herbert Boyer detailing recombinant deoxyribonucleic acid technology stands as a landmark achievement in the history of science. Their work not only provided a powerful new tool for biological research but also laid the foundation for the modern biotechnology industry. While ethical considerations and societal concerns continue to surround the technology, its impact on medicine, agriculture, and our understanding of life itself is undeniable. The principles and techniques outlined in their seminal paper continue to be refined and expanded, paving the way for future innovations in gene editing, personalized medicine, and sustainable agriculture. The enduring legacy of Cohen and Boyer's work lies in its transformative power to unlock the secrets of life and to harness those secrets for the betterment of humanity. The recombinant deoxyribonucleic acid technology described in their 1973 paper not only revolutionized basic science research but also transformed the landscape of medicine and biotechnology. It represents a quantum leap in our ability to manipulate and understand the genetic material of living organisms. This conclusion encapsulates the overall evaluation of the work's claims reaffirming its enduring significance and highlighting its transformative impact on science and society. The technology has truly helped to unlock many secrets of life.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.